Your CO2 levels, your plasma levels, oh yeah, you still yeah. Right.
All right, let's go ahead and come back together here. Just real quick back on the Notebook LM, I forgot to say how to access that if you haven't figured that out yet. If you're logged into your Point Loma Gmail account and you just open up a new tab in Chrome, there's that little spot on the right that has all the little dots, you know, where you can access the different Google things. It's just one of those. You just click there and then you scroll down to the bottom and you'll see it there. So, okay, so for this question, so we've got somebody who has had chronic diarrhea. What are we losing with diarrhea? Say it again, Tiffany, go ahead. A little bit louder? No, okay. I'm sorry, I said the wrong name. Yeah. What's your name again? Huh? Yeah, that's what I said. I said Tiffany. Sorry. Yeah. I had it right. You looked like you were going to answer. What were you going to say? Acid. You were losing acid. Okay. Anybody else? Definitely losing water for sure. Pancreatic juices, which are very alkaline, right? And so we, so with vomiting, we lose a lot of acid, right? With diarrhea, we lose a lot of alkaline or basic fluid. So this is one of the causes of metabolic acidosis, right? Excessive diarrhea, excessive vomiting would be a cause of metabolic alkalosis, right? This is one of the causes of metabolic acidosis. So if we're in metabolic acidosis, what are a couple ways the body can try to correct for this? Go ahead. Okay, through what part of our body? Urination, okay, yeah. So the kidneys can get rid of more hydrogen ions for sure. Okay. What else can the kidneys do? Yeah. Perfect. Hold on to bicarbonate. And then the other one, we see it happening here, right? One of the symptoms is rapid breathing, right? So with rapid breathing, we're getting rid of a bunch of CO2. And if we get rid of a bunch of CO2, then I'll write out the equation for this, right? So if we've got H plus HCO2 3 negative. And then we've got on the other side of that, so we can write this out as a reversible reaction, right? H2CO3. Okay. And then H2O and CO2. Okay. So in this case, so I'd want you to kind of know which way that this equation would push in order to, yeah, with the situation we have going on, right? So when we have a bunch of extra hydrogen ions, right, it's the base that needs to help us with that, right? So it's bicarbonate that needs to help us with that. So the hydrogen ions are going to bind to the base here, which then becomes the acid, the carbonic acid, okay? And then that can dissociate into water and CO2. And if we're breathing a lot, then we can get rid of this CO2, right? We'll breathe that out. And then it makes it so we can't drive the equation back the other direction, right? We can't do that without that CO2 available. We can't push it back the other way and then remake that hydrogen ion, right? So it's helping us get rid of hydrogen in a way, right? Without actually getting rid of hydrogen, but it makes it so we can't reform it. So really, we would want to know that the equation is not reversible here. I mean, it is, but in this case, it's moving to the right, right? Okay. Questions on that? Yeah, go ahead.
Both do the essence factor, so dehydration.
Yeah, are you talking about the H2 over here? Yeah. I don't think it's a significant enough amount of water to affect that, but I like the way you're thinking. Yeah. Other questions or thoughts on that? Okay. All right. Well, let's move into some muscle physiology then for today. So we're going to kind of just run through a review slash overview of all the steps related to muscle contraction here. Okay, so we're going to start with the activation of the neuromuscular junction, kind of that first part of connecting the nervous system to the muscle, the skeletal muscle. And then we'll talk about how we activate the skeletal muscle to turn it on, get it ready for contraction, and then cross-bridge cycling. So I'm just going to open up kind of a blank whiteboard and just kind of write out these steps. Let me do that here real quick. And you guys will probably want to do the same. Okay, so we've really got kind of three different processes that we'll talk about. And that first one is just the neuromuscular junction activation. I'll try to write slow so I don't write too terribly, but please stop me and ask me if you can't read one of the words that I wrote. Okay, so neuromuscular junction activation. I'm going to abbreviate action potential as AP. Okay, so we have an action potential that's going to travel down a motor neuron. Okay, till it gets to that neuromuscular junction, NMJ. Okay, we'll talk about the details of an action potential and how it starts next. Starting next week when we get into the nervous system. So we don't have to know those details yet. But that action potential goes down, gets down to the end of the neuron there, and when it gets to the end of the neuron, it leads to. So we'll just call that step one. Step two, it leads to. So action potential causes calcium influx into the neuron. Okay, so we get calcium. That's just Ca with two positives, right? Plus two. Okay, so that calcium flows into the neuron. So there's voltage gated calcium channels on the end of our neurons that are the shape gets changed when the voltage changes, right? So that opens them up, and so then calcium rushes into the end of the neuron. Okay, just like we were talking about last time, this is one of the primary cell signaling mechanisms is utilizing that calcium gradient, right? We got a lot of calcium outside of cells, so if we just open up the channels for calcium to go in, calcium goes in there and changes some proteins, right? Changes the shape of some proteins. kicks off a chain of events that leads to acetylcholine, we can just abbreviate it as ACH, release. Right, so acetylcholine is just stored in vesicles in the end of our neurons, right? And so with the influx of calcium, that activates a chain of events that then makes those vesicles fuse with the end of the neuron, right? And open up their contents into that neuromuscular junction. Really, right? Actually, like into the synapse there, right? So that space, or the synaptic cleft, we could call it, right? Into that space. Okay, so then acetylcholine binds to acetylcholine receptors on the sarcolemma. Sarcolemma is a muscle cell membrane, skeletal muscle cell membrane, right? It has a bunch of acetylcholine receptors on them. Okay. When acetylcholine is bound to the receptors, that leads to two things: influx of sodium, as well as an efflux of potassium. So this is like not a 100% specific cation channel, right? That allows both these to flow through. But when we talked about this last time, right? When sodium can move and potassium can move, which one's going to move more? Sodium, right? Because it's going with the electrical gradient and the chemical gradient, right? So we get a lot bigger influx of sodium than we do efflux of potassium, and this leads to depolarization. Okay, so this would be the acetylcholine receptor, and the activation of the neuromuscular junction here is an ionotropic mechanism, right? We talked about this last time, where there's the metabotropic, right? Metabotropic is more like the second messenger system. There's multiple steps that happen after that first messenger binds, right? In this case, we have the acetylcholine binding. It's the same channel that or the same protein that was the receptor is also the channel, right? So it just binds to it, opens it up, the two ions can move, right? So that's an ionotropic mechanism, right? We're just changing the ion permeability there with that protein. Okay, so we got depolarization. This is known as a graded potential. We'll talk more about different action or different potentials when we get to the nervous system, but this is not an action potential yet. It's a graded potential, right? Graded potentials are these little local changes in membrane potential that kind of spread a little bit. Okay, if we get enough stimulus, then those graded potentials can then make their way to the regular part of the muscle cell membrane and eventually cause a action potential. So action potential on muscle cell. Okay. So then we're kind of shifting to what would be called excitation contraction coupling. So excitation contraction coupling is really the activation of the skeletal muscle cell. Okay. And yeah, it's really kind of starting with the step above, step number 6, right, where we're getting that action potential. Okay. And there's one really unique thing about skeletal muscle cells is that they're huge, right? They're massive compared to other cells, right? They're about 100 times thicker than a red blood cell, right? On average, right? And they can vary in size a lot. And so what that means is if we just had the action potentials kind of spreading on the outside of the muscle cell, those action potentials wouldn't really have any effect on the deeper parts of the muscle cell. And so we need to have some mechanism to allow that action potential to spread deep into the muscle cell. And so who remembers what that structure is called that allows for that to happen? What allows for action potentials to go deep into a muscle cell, skeletal muscle cell? Yeah, talk to each other real quick on that. Check in with each other. Okay, what's that structure called? T-tubules, yeah, transverse tubules, right? So T-tubules, um, so pretty much it's part of the membrane still. They're called invaginations of the membrane, right? It kind of just like pokes inwards, and that allows the action potential to dive deep into the muscle cell membrane. We'll have there's some images on the slides of these things coming up in a sec too. But that allows for the muscle cell or the action potential to go deep into the kind of depths of the muscle cell. Okay, so it goes down the T-tubules. When it's going down the T-tubules, it's going to activate the sarcoplasmic reticulum. Right, so there's a bunch of different sarcoplasmic reticula as we go deeper through the muscle cell, right? And so that action potential is by going down the T-tubule is going to activate a bunch of them, right? Um, and what does the sarcoplasmic reticulum do when it's activated? Releases calcium. Okay, so again, here's calcium running one of our molecular mechanisms inside of our cells. Okay, so sarcoplasmic reticulum releases calcium, and then that calcium, which protein of all those proteins you guys looked at in the pre-class video, does it bind to? Troponin, yes. Calcium binds to troponin. Okay. And then we get a conformational change of troponin, right? We got all these amino acids strung together that made up troponin, right? Um, then the shape of that molecule is dependent on the way those amino acids interact with each other, the way the amino acids interact with the water around and the pH and other things, right? So if we have this ion that has a positive 2 charge binding to one of those amino acids, right? Now it's different, right? Now that amino acid is different than it used to be. So that means it interacts with everything around it differently in such a way that it totally changes its shape, right? So troponin totally changes shape. And troponin, so binds troponin and changes shape. Okay. Any words you can't read on here before I keep moving on? Or up here. We good? Don't be embarrassed to ask. I'm not offended. Okay. All right. So troponin changes shape. Troponin is attached to another one that's blocking the binding site for the myosin head to bind to actin. And that's the one that actually has to move. What was the name of that protein? You guys remember that one? Tropomyosin, we got that. So troponin then moves tropomyosin. All right. Now we got an open actin myosin binding site. So that's really the end of excitation contraction coupling, right? The muscle cell is now ready for contraction. Okay. And then we move on to the last part, which is cross bridge, bless you, cycling. Okay, so cross bridge cycling is our last part. This cross bridge cycling is a cycle, as the name implies. And so we can kind of start in different places, but we'll start with we'll start with the myosin head attached to actin. We'll just start there, okay? And it has a spent ATP on it, so it we'll just say the ATP is already spent. So ATP already used, okay? So the next step here in the cycle would be we get a new ATP that comes. New ATP, okay? We're going to say what that does is phosphorylate the myosin head. Okay. There's a special enzyme that helps us do that, helps us break down that ATP. What's that one called? It's an ATPase, and it's myosin ATPase, right? So myosin ATPase is what kind of does this, right? It breaks down that ATP, takes that phosphate and kind of lets it bind to the myosin head. And just like the calcium changed the shape of troponin, the phosphate is going to change the shape of the myosin head, right? And this is a pretty big movement too, in molecular terms. It moves the myosin head quite a bit. But while it's doing this, it breaks pulls the myosin head off of actin. Okay, so this new ATP is really what's detaching head from actin. Okay, and so kind of all in one fall swoop, where that myosin head's coming off of actin, it's getting phosphorylated. The myosin head's getting phosphorylated, and it's cocking the myosin head. Okay, so cocking myosin head. It's what's called putting it into its power-packed position. Right, so we're taking that myosin head from its kind of spent position and put it into its power-packed position, but we're doing that when the two aren't touching each other, right? Okay, and this is always the kind of the interesting part is that we're using our energy, we're using our ATP when the two aren't touching each other, right? So it's similar to cocking a gun, right? When we put our energy into the gun, but that's not when the bullet comes out, right? We cock it, right, and it's ready to go, right? But then we have to release that in order for the bullet to actually come out, right, and for it to fire. So it's similar with this. We're cocking it while they're detached, okay, and then it can come back up and attach again. So reattach. Myosin to actin. Okay. And then finally, with the release of inorganic phosphate, that causes the power stroke. And muscle shortening. Okay, so it's the dephosphorylation of the myosin head that makes it go back into its position, right? Which would be, but now it's attached to actin, right? So it's attached to actin and it springs back, so it brings actin with it, right? So that's kind of the key there. And now we're back to where we were, right? We're back to starting right there, and we can start that cycle over again. Yeah, go ahead, Tim. Number 5 says release of PI, like inorganic phosphate. Okay, you could just say phosphate. That causes the power stroke, which is what causes the muscle to shorten. And I just put the arrow back up to number 1 because we're kind of starting the cycle over again from there. Okay, go ahead. Yeah. It does as well. Yeah, yeah. And there's kind of some debate around this, like what actually causes the power stroke. It's probably the phosphate coming off, but they do kind of come off around the same time. And so, yeah, good question. Yeah. Other questions on that? Go ahead. Yeah.
Does the cross stroke, does it just keep happening, or does it need one like excitation, contraction, coupling in order to just do one motion?
Great question. Yeah, it happens thousands of times within a matter of a minute for sure. So it's just as long as calcium is present and as long as ATP is present, it's just going to continue happening really, really rapidly.
You just get like one potential of the synapse, it will lead to like
No, so actually, we're getting same thing, thousands of action potentials that are arriving at the neuromuscular junction, which leads to this steady high frequency action potential on the muscle cell membrane, which then that leads to a steady activation of the sarcoplasmic reticulum, right? Which then that leads to just a steady flow of calcium getting pushed out, so that calcium levels stay high in the muscle cell, and then it's more likely to stay bound to troponin, right? As soon as we start decreasing that activation, then calcium release slows down, and so then we activate. We'll talk about in a second, but we activate these pumps that start pulling calcium back in. And then that's what will eventually stop the cross-bridge cycling. Yeah. Yeah.
Sam, a specific mechanism, like just explain cross-bridge cycling, or would you want the whole, all three of these mechanisms?
You need to know them all. So yeah, it's good to know how they connect too, for sure. So yeah. Mhm. Okay. Other questions on that? Okay. So let's go back to the slides here. So yeah, we kind of just went through that. Okay. Let's just look at a couple images that kind of help solidify that. So here's kind of our early part activating the neuromuscular junction, right? So we got our action potential going down the muscles or the neuron, right? It's going to activate these voltage gated calcium channels. Voltage gated calcium channels change shape, open up, calcium rushes in, and then that calcium is going to activate all these little vesicles of acetylcholine. They just fuse with the membranes, like we were talking about last time, that exocytosis mechanism, right? They're going to fuse with the membrane, dump the acetylcholine out. Acetylcholine is then going to bind to its receptors, which then allows for both sodium entry and potassium exit, but sodium entry dominates, right? So we get a local depolarization, right? That's that graded potential right there. That local depolarization, if it spreads enough to the regular part of the muscle cell membrane, where the voltage-gated sodium and potassium channels are, then we get an action potential, right? So then we propagate this action potential down the muscle cell membrane, okay? We'll see those T-tubules in a sec, but questions on this? I guess this one, how does it stop? How does the signal stop? As soon as we stop sending action potentials down, then we stop sending calcium in, so then we stop releasing acetylcholine, right? So that's part of it. But then we also have enzymes, one special enzyme called acetylcholine esterase. Sorry, I could do better at spelling that. Let me try it again. Esterase. So acetylcholine esterase is an enzyme that just breaks down acetylcholine. So that's floating around in the synaptic cleft as well. So that's going to be breaking down the acetylcholine that's out there. And that will stop the signal if we're not continually releasing more, right? So the action potentials are stopping, which makes us not release more. And then we're also breaking down acetylcholine. And that's what this is. Yeah, I guess I didn't need to write it because it was right there. But acetylcholine esterase right there. Okay, questions on that? Yeah, go ahead. It's constantly working. Yeah. So it's just there. It's doing its job. It's just a matter of concentration of acetylcholine, right? So it's when we stop, when we slow down the release of acetylcholine, then acetylcholine esterase will kind of eventually dominate and decrease the level of acetylcholine. Yeah. Okay. Other questions? Okay, so a couple things that can happen in the neuromuscular junction, pathology-wise, botulism toxin, or botulism, which is caused by botulinum toxin. The real mechanism here is that it's blocking acetylcholine release. When we look at the nervous system, we'll look a little bit more in detail what's going on with the mechanism there. But in the simple way to look at it is that acetylcholine can't be released, and so this is what leads to the paralysis of muscles, right? So same thing that we have in facial injections and things like that. That for wrinkles, but also for headaches and things like that in the neck, right? It's just blocking those muscles from activating, and so yeah, it inhibits them. Myasthenia gravis is a autoimmune disorder. Oops. And it attacks, the immune system attacks the the receptors, the acetylcholine receptors. And it leads to destruction of the acetylcholine receptors. Right? And so if we destroy our acetylcholine receptors, we can't then contract muscle anymore. There's two primary types of myasthenia gravis. The type 1 myasthenia gravis is only affects the extraocular muscles in the eyes, and that's the more common type. And so, yeah, people just lose the ability to move their eyeballs. But then the more extreme type is when it affects all muscles, skeletal muscles, including the diaphragm, right, which can be potentially life-threatening in that case. Okay. Questions on that? Okay, so just a couple things here, just to kind of solidify this. We've got calcium is obviously playing some key roles here. So this is showing the T-tubules, right? So this would be that muscle cell membrane and the T-tubules going down in. You can see that the T-tubules are directly connected to the sarcoplasmic reticulum via a certain protein there. That image on the top doesn't show it that clearly, but we can see over here a little bit more clearly, we have two specific types of receptors. Okay, so we have these dihydropyridine receptors, and then we have ryanodine receptors. Okay, we'll start with the first question though. Which troponin does calcium bind to? There's a couple different types of troponin, right, that you guys saw in the book. So troponin C is the one. All right, easy to remember because it's calcium one. Okay. And so when it binds, it moves this blue tropomyosin, right, from blocking the site to now having those sites open. Okay, so we got that part. And then if we kind of zoom in here, it's kind of hard for me to see, so I'm guessing it's probably kind of hard for you guys to see too. Let's zoom in on that image a little bit better. Okay, so we've got these dihydropyridine receptors. Those are the ones that stick out into the T-tubule space and in that muscle cell membrane. Okay, and those are directly connected to these ryanodine receptors. And the ryanodine receptors are the ones that we need to activate the sarcoplasmic reticulum and cause calcium to open up. Okay, so they're both, the dihydropyridine receptors are voltage-gated receptors. So the voltage changes their shape, and they're directly connected to the ryanodine receptors, which when they change their shape, that changes the shape of the ryanodine receptors, and calcium influx can happen. Right, so that's really the key part there in understanding. So this is to change shape in response to the action potential. Okay, sorry, that's kind of small, but the dihydropyridine receptors change shape in response to the action potential going down those T-tubules. Okay, and then those then the ryanodine receptors are opened by these dihydropyridine receptors. Okay, so we just have that direct connection between our muscle cell membrane and the sarcoplasmic reticulum. We'll see in the cardiac myocytes in just a minute that we don't have that same mechanism there, and it changes the way that the cardiac cells contract versus the way skeletal muscle cells contract. Okay, questions on that? Okay, so cross-bridge cycling. This is just an image that shows the steps of cross-bridge cycling. I want you guys, just as like a little review of what we just talked about, to kind of interpret these images, write in what's going on, talk about with your neighbor what's going on, and yeah, for right here, for each one, write in what's happening with ATP, calcium, myosin head, troponin, actin, etc. Okay, so just kind of talk through what's going on, and we'll come back in a sec and fill that in. Okay, go ahead.
Yeah, so you can start. I think so.
Yep.
It's changing the shape, and then
Yeah.
would it be for the release of calcium or the influx of calcium?
Release. Yep. We
Here.
don't rely on any,
In,
yeah, we don't rely on extracellular calcium in skeletal muscle cells, just the stuff that comes from the sarcoplasmic.
so the opening of the renin receptor would allow the release of calcium.
Yep, exactly. Yep.
Thank you.
Mhm. Yeah.
Here too, isn't ATP technically, because that's ADP plus a phosphate?
Going here, so they're detached from each other at this point.
Okay.
It just, went from here to here, so this was like the breakdown of ATP, which caused the break apart of actin and myosin, yeah. So yeah, they're not bound to each other, but they're both still bound to the myosin head. Yeah, so both connected to the protein, just not to each other anymore.
Which then causes like the so then when it's so much, that's when it's basically the attention. In the little
Yes. But it's something like. It's spent.
Yeah. Except we're not. It's starting to accident. All of this.
Yeah.
You're good, yeah.
We were wondering,
Yeah.
since it stays in like a 45-degree angle for the time, the only the only thing that happens is the binding of ATP for it to kind of go back to the 90-degree.
Yeah, that's that's the phosphate binding to the like the phosphorylation of the myosin head pretty much is what causes that shape change. Yeah.
But after it power strokes, it can't be like that until a new ATP comes in.
Yeah, exactly. And it'll stay attached as well. Yeah, the myosin head will stay attached to actin and it'll stay in that 45-degree angle position until the new ATP comes. Yeah. Yep, exactly. Yeah. Mhm. All right, guys, let's go ahead and come back together here.
Like that already.
Okay, so where did we start when I was writing it out? What did you guys say? Did you anybody try to do that? Where was the starting point versus what I was writing here? I said the myosin head attached to actin, the ATP is already used. So which one would that be? Top, the very top, what are you thinking? Yeah, very top probably. Yeah, I agree with that. Yeah. So let's call that number one just so we can talk about it. Okay, 2, 3, 4, 5, and 6. Okay, so yeah, so with number one, we've got the myosin head still attached to actin, okay, and it's at the 45-degree angle, okay. So then from 1 to 2, right? The new thing is the ATP comes, right? The new ATP. What happens if no new ATP comes? Rigor mortis, right? This is what causes rigor mortis, right? When we stop breathing, stop making ATP, myosin heads are just stuck on actin and they're not moving, right? So our muscles are very stiff in that case, right? So that new ATP comes, okay? And then difference between here and here, we just see that ATP is becoming ADP and phosphate. So the myosin ATPase did its job, broke down ATP into ADP and phosphate with that hydrolysis reaction. Okay, and so then we see kind of that detaching there, right? That's the big difference there. That new ATP comes, so the function, that first role of the new ATP coming is to detach the myosin head from the actin, okay? And then the phosphorylation of the myosin head causes this to go from that 45-degree angle to the 90-degree angle, so we're changing shape of that myosin protein, okay? What else is happening there in number 4? It changed shape and it did what? Yeah, it bound to the actin, right? The myosin head bound to the actin. So now we're reattached there, okay? And then we see the removal of the phosphate, which that's what causes the power stroke, right? So number 5 is the power stroke, okay? And so that's really where the slide-in would happen in the sliding filament theory, all right? So that's really where the muscle is shortening right there in number 5, okay? And then eventually the ADP comes off as well. And then we're back to where we started. Okay. Questions about any of that? All right. Yeah, go ahead.
The myosin ATPase is working in between step 2 and
Yep, exactly. Yeah. Mhm. Other questions? Yeah, go ahead. Mhm. Exactly. Yep. It will not detach itself until that new ATP comes. The myosin head will not detach until the new ATP comes. Good questions. Other questions? Go ahead. So that would be going from step 3 to 4 in there. Yeah. That's kind of where the cocking happens. Other questions? Okay, let's just talk briefly about muscle relaxation. Um, and so this is just kind of a nice like flow chart from your book. Yeah, so the steps in contraction are there on top in green, but then the steps in relaxation are there in blue on the bottom. And so when we stop sending action potentials or we slow down the frequency of action potentials sent to our down that motor neuron, then we stop releasing as much calcium, right? And so really what's happening here is calcium is pumped back into the sarcoplasmic reticulum, okay? So we get a decrease in calcium concentration. And if we don't have as much calcium around those troponin molecules, then calcium starts coming off, right? We don't get calcium bound to troponin anymore. So it releases from troponin, right? And then we cover up that binding site again. Okay, we have a special protein that's bound to our sarcoplasmic reticula, which is pulling calcium back in. We talked about this one a little bit last time. It's known as that SERCA. Okay, it's sarcoplasmic or endoplasmic reticulum calcium ATPase. Okay, so it's just pretty much using ATP to pull calcium back into the sarcoplasmic reticulum or in other cells that it's not a sarcoplasmic reticulum into the endoplasmic reticulum. That's why it has those two different names. What type of transport would this be? Talked about it, the different types last time, right? What is it? Primary active, perfect, yeah. Primary because of the ATP, active because it's going to keep pushing calcium in even when there's already more calcium inside the sarcoplasmic reticulum. Okay. All right. Questions on any of this? Okay. How are we doing? Do we need a 5-minute bathroom break? Are we okay? Think we're good? Okay. I haven't taught this class in a shorter format for a while, so I'm like, do we need breaks still? I'm not sure, but I don't need a break. You guys want to keep going? Okay, let's do it then. All right. Okay, so kind of shifting gears a little bit to a little bit more kind of practical outside of what we're just talking about. One concept in our skeletal muscles called the length tension relationship. You guys probably remember some of this from your undergrad physiology. Length tension relationship pretty much means that a muscle is going to be its strongest somewhere in the mid-range of its range of motion. Okay, so the easiest example to think about is like with your biceps brachii. Okay, if you try to do a curl out here, it's not going to be as strong. If you try to do a curl right here, you're not going to be able to produce as much force. If you're doing right here, you can produce the most force. Okay, and there's the kind of reason for that has to do with our sarcomeres. Nope, not visible body. Yeah, it has to do with our sarcomeres right here. Okay, so here's our myosin with all the little heads sticking out, right? These represent the actin. Okay, these numbers correspond with the spots here. So 1 is actually over here, right? 5 is right there. Okay, so when a muscle is all the way lengthened already, that would be number 1 here. Okay, so this is talking about specifically a sarcomere. When a sarcomere is all the way lengthened, we have all of these myosin heads that aren't touching actin, and so they're not helping us produce force. Right? They're just kind of swinging around in cytoplasm. They're not doing anything. Okay. And then on the opposite end of that, number 5, when our sarcomeres are all the way shortened already, there's just no more room for them to shorten. Right. So number 1 would be when your elbow's all the way extended. Okay. Number 5 would be when your elbow's all the way flexed. Okay. And so when your elbow's all the way flexed, much larger majority or proportion of your sarcomeres look like this already. So those ones aren't helping us produce force, right? There's probably still a few that have some ability to shorten some, right? But we're relying on less sarcomeres at that point so that we can't produce as much force, right? So this is the force, the tension that the sarcomere can develop at each of these ranges of motion. So not much here at 5, not much here at 1. And then we've kind of got this sweet spot in the middle, right? Where we have good overlap of actin to myosin, meaning that we're using a lot of the myosin heads, but there's also still room for the sarcomeres to shorten, right? So we got some room, and we're using a lot of the myosin heads. Okay, so we'll say optimal overlap of actin, sorry, and myosin, and still room to shorten. Let's see. We got some room to shorten, and there's optimal overlap there. Okay. This is something that we kind of naturally know, right? When we want to jump high, we're not going to jump high with our legs all the way extended, right? We're also not going to put our butt all the way on the floor and jump high, right? I was watching I was at a Padres game recently when Tatis robbed Rafael Devers' home run. How many people know what I'm talking about? A couple of you? Okay, well, I got a video here so you can all see it. But I was actually sitting in right field for that game, not right behind Tatis. I was up in the upper deck, but this was like, I think it was just the first inning maybe, and Rafael Devers can hit a lot of home runs. So here it is. Let's watch this. Let's watch how Tatis. That's really loud. Yeah, so I'm gonna pause it there. So watch what he does. He goes down, right? And then he comes back up, right? There it is. Yeah, incredibly impressive. Watch. The part I want to focus on is this, right where he loads up, okay? He's not going to jump with his legs extended. He's going to see how he loads down right there. He comes down like this, right? Before he jumps up, right? So he puts himself into a position where he can produce a lot of force. And he jumped really high, right? Probably 4 feet high. Yeah, he's an incredible athlete. And he just sat there because he's because it's athletes. Yeah. I think he's had eight robberies or something like that this season. It's been incredible. But yeah, so there's that's the length tension relationship, right? Because he didn't jump from here. He went like this and then he jumped up, right? And also the other thing that he's really activating there is a stretch reflex, which we'll talk more about next time we get to the nervous system. But also important to stretch your muscles first to activate the stretch reflex, and then that helps you jump too. But yeah, go ahead.
Correlate the graph to when the muscle is the strongest, or is it kind of like a mix?
Yeah, probably both 2 and 3 right in there. Yeah, so somewhere just in that top range, just where that tension development is closest to max there. Yeah.
Isometric contraction, could you argue that 5 would be the strongest as you don't really want the muscle to shorten more, but just to maintain?
It's a great question. No, so we yeah, because and you could really do it with 3 different isometric contractions with your biceps. Like you could push from right here, right, push from right here, and push from right here, and you would most likely produce the most force at this point right here if all 3 were isometric contractions. Yeah, yeah. And I used to usually in my undergrad class, I bring a dynamometer in and do that exact thing, like have somebody who thinks they have big biceps come up and push on them with the dynamometer, and we measure the force production. And yeah, the one in the middle is typically always the one that does it. Yeah. Okay. Other questions on that? Okay. All right. Nope, we don't need to watch that again. All right. So another related concept to skeletal muscle contraction is the 3 types of contractions. And we just got into that a little bit with isometric, right? So we've got the 3 types. So isometric, oops. And isometric is no movement, right? We have concentric, which is shortening. And then we have eccentric, which is lengthening. Right, so those are kind of our 3 types of muscle contraction. And so the example I always use is a bench press, right? So if we have somebody doing a bench press here and we just isolate those 3 types of movement, we can kind of try to figure out which one we could lift the most weight with, okay? So this one, if we kind of help the person take the bar down to their chest and then they just have to push it up, what type of contraction would that be? Actually, don't answer it yet. Yeah. Well, we'll go. So that one is that one. The middle one is isometric. Sorry, now I'm giving it away. Middle one's holding still, and the third one is all they have to do is lower it down. Just check with each other real quick on the first one and the third one, since I already gave away the second one. Just match those up with the type of contraction they are.
Well, I think it's like looking at.
All right. So what do we got? So what would we call that? The first one, the up slowly one? Which type of contraction? Okay, we're a little mixed between concentric and eccentric. We can choose a muscle if that makes it easier. They're all pretty much doing the same thing, but that's okay. We can choose a muscle. Let's choose the triceps. Okay. So when we're going up slowly, what are our elbows doing? Extending, and so our triceps are shortening. Yeah. So that would be concentric. Okay. All right. Holding still, we already said was isometric. So then just going down, we can choose the triceps again. We're starting up here. We're letting the bar go down in a controlled fashion. Our triceps are lengthening when we're doing that, right? So that would be the eccentric type of contraction there. If you were just to do this with the maximum amount of weight that you could with each of them, which one do you think you could put the most weight on the bar with? Probably the eccentric one, right? Probably lowering it down. And it might feel like it's kind of cheating because you're using gravity and stuff, but your muscle is doing the work to lower it down slowly, right? It's still doing that work. And so it's just known that with eccentric contractions, so eccentric is the strongest. Eccentric contractions can produce about 150% of the force that concentric and isometric can. And so when you strain a muscle, for the most part, it's going to be during the eccentric phase of contraction. So it's usually with landing, right? You're landing from a jump, it's that eccentric phase, right? Or you're going to push off, but you load first, right? Just like Tatis did when he went down, he loaded first. That would be where he would strain it most likely, is when he's coming down, not necessarily when he's coming back up. In the concentric phase is when he's down and then he's coming back up, right? That would typically be where you get it, because you put a lot more stretch and strain on your muscle during that eccentric phase. And the interesting part is, there's a lot less activity during, less electrical activity during eccentric. Meaning that if we put an EMG on somebody's muscle, when they're doing these three types of contractions, so EMG picks up like the level of electric activity in a muscle. If we put that on the muscle during these three types of contraction, we would see the least electrical activation during the eccentric phase. So we're really relying a lot on just the intrinsic components of muscle, those titin proteins that we talked about that are shaped like a spring that stretch out really well. And when they stretch out, they want to bounce back, and that gives us the force, right? And that's what Tatis is doing when he loads down like this, is he's stretching out that titin, right? And then he gets to accelerate a lot higher because that titin molecule springs back and gives him a lot more force to do that, right? So yeah, questions on that? Okay, one more thing related to skeletal muscle, and then we'll move on to cardiac and smooth muscle in the end here. So motor units. A motor unit is defined as one motor neuron and all of the muscle fibers or cells it innervates. Okay, so one big motor neuron comes out of the spinal cord. It goes down and branches into a bunch of little branches, and it connects to a bunch of skeletal muscle fibers or cells. Okay, so one motor neuron and all the muscle fibers or cells, same thing with muscle, that it innervates. Okay, so like, for example, here, these are all color coded, right? We see kind of the orangish, reddish one coming out. It goes down and connects to all of the orange or red muscle cells, right? One thing with this is all of the muscle cells are going to contract at the same time within that motor unit. Okay, so that's what this all or nothing is that when we send action potentials down motor unit number 1, all of the cells connected to motor unit 1 have to contract. We can't just like pick and choose ones within it. We're just sending the action potential down those. Okay, so that's the all or nothing phenomena. And what we find is that within that one motor unit, we have similar types of muscle fibers. They're all going to be similar as far as their oxidative capacity. If they might all be type 1 fibers, they might all be type 2A fibers, all within that same motor unit. Okay. And then the other thing that we find is the size principle, which means that we're going to contract the small ones first then up to the medium ones, then up to the large ones, when we're trying to produce more and more force, right? So we just need a little bit of force, we're just gonna use the small ones. If we need a little bit more force, we're gonna do the small ones first, then we're gonna kick in the medium ones too, right? We still need more force, we're still gonna use the small ones and the medium ones, but then we're also gonna kick in the larger ones, yeah.
What's small, medium, large?
Great question. Yeah, it's a combo of both. So it could be within a small motor unit, you're going to have a small number of muscle fibers, and those cells are going to be pretty small. Right? As we get bigger, we get more cells, and the cells get bigger too. So, yeah. Okay. Questions on that? Other questions? Okay. Okay, let's move on to the other two types of muscle cells. Let's talk about cardiac myofibrils or cardiac muscle cells. So as far as just kind of the cell itself, they're very different than skeletal muscle cells. They're branched, which is interesting, right? We can see a single cell right here. You can see how it branches into two different spots, right? So one side of it is just singular, but then the other side branches. Okay, so that's what branched means. The gap junctions, we talked about these last time, right? So in the little spaces between the cells, we have little holes, right? And they're shown kind of right here. Those little spaces between the cells are called intercalated discs. And within those intercalated discs, we have gap junctions. We also have desmosomes. Talked about those last time too, right? Those rivet-like structures that hold the cells together. Gap junctions are there to allow the signal to pass from one cell to the next, right? It's just like a hole between the cells, right? And so those are the primary things we find there. Yeah, and other thing about cardiac muscle cells that makes them different from skeletal muscle cells is just packed heavily with mitochondria. So lots and lots of mitochondria, very, very oxidative cells. Makes sense, right? We don't want these guys to fatigue, right? It's not in our best interest if these fatigue, right? They have a single nucleus instead of multiple nuclei like skeletal muscle cells because they're not super long and big like skeletal muscle cells, and they contract all in together, which is a funny term, and I put the pronunciation in there for you. It's called syncytium. Syncytium is when, really, if you think about it, when that action potential is going to start, or that signal is going to start up on our SA node, up in the top part of our heart, right, but up by our atria. We want to send that signal down. We don't want the top part of our atria to contract first, and then the middle part, and then the bottom, right? We want it all to contract in one, so we get a nice strong pump, right? So the gap junctions help with that, and then the top ones are kind of waiting just a little bit, and then they all contract together with those gap junctions. Same thing in the ventricles, right? And as we go down the ventricles, the signal travels faster and faster and faster as we go down, so they're all contracting together. Yeah, so that's just a little bit about how they're different. We still have T-tubules, we still have a sarcoplasmic reticulum, but a couple of the differences there. The other big difference that I already mentioned is we don't have the dihydropyridine receptors in cardiac muscle cells. Okay, so cardiac muscle relies on extracellular calcium to then induce the sarcoplasmic reticulum to release calcium. Okay, so we get depolarization going down the cell membrane, just like we did in the skeletal muscle. Okay, instead of having a dihydropyridine receptor, we have what's called an L-type calcium channel. We'll talk about these more when we get to the heart. These are, the L stands for like long lasting. Okay, so an L-type calcium channel gets opened up because of that depolarization. That lets extracellular calcium come in. That extracellular calcium activates the ryanodine receptor on the sarcoplasmic reticulum inside those cardiac myocytes, and then that's what leads to calcium influx. Okay, so this is just a little bit slower of an activation process, which is actually helpful in the heart. We want to let the heart have some time to build tension, right? It's not like our skeletal muscle where all of a sudden we need to turn and run immediately, right? We're waiting for the time between beats for the heart to fill up, right? We're waiting for the top part of the heart to contract before the bottom part does, right? So there's some built-in kind of slowness to this, which is helpful, right? And we need that. And this also gives us some variability in how strongly we contract the heart too. We'll talk more about that when we get to the heart as well. But how much calcium flows in is going to dictate how much contractility we produce, right? And that's going to be changed by the sympathetic nervous system. So we can kind of vary that a little bit more here. Okay, and so the metabolism of our heart, what does our heart use for energy production? It uses mostly lipids, definitely some carbs, ketone bodies, and amino acids if we need to. But the interesting part is it's 100% aerobic, right? The heart really doesn't do anaerobic metabolism unless it's forced to. It can if it's forced to. When somebody's having a heart attack, you see that happening. But for the most part, we don't really use any anaerobic metabolism in the heart. Okay. All right. We haven't talked about the other action potentials yet, but we will coming up soon. But we'll talk about the cardiac ones here because this is where the main point we're talking about these heart cells. When we look at a little graph like this, we'll look at this for neurons and for muscle cells, skeletal muscle cells as well. When we look at this voltage graph here, we can see resting membrane potential, okay, is right here. We can see with B, we have a big depolarization, okay? This big depolarization is in all cases where we're going to talk about this caused by sodium influx. Okay, so B is caused by sodium influx. Okay, C is going to be caused by closing these sodium channels. Okay, and then a little bit of potassium efflux. Okay, we'll look at a bunch of different types of action potentials throughout the next month or so. But usually the repolarization when we go back down towards resting membrane potential is always going to be caused by potassium efflux. But it's important that the sodium channels close too, or else we would have kept going up, right? So sodium channels close, potassium starts to leave a little bit, but that also leads to calcium influx. Okay, so that's D is a unique part of the action potential here with calcium coming in, and it leads to this big plateau phase, right? So the plateau phase in a cardiac action potential is caused by calcium coming in. Okay, and then finally, when those calcium channels stop, when those calcium channels close, so calcium closes, right? But now the potassium efflux that was happening can kind of take over and in E and lead to repolarization. Okay, the nice part about this graph that I like is that it doesn't just show the voltage change, it also has a tension graph right here in the heart. Okay, go ahead. You can ask a question first.
The tests, would we need to be expected to draw these things? Or is it ourselves?
Not draw yourself because it is on the computer. You don't really draw, but you would need to know, be able to label them and know what's causing each stage and kind of where we've talked about it, the importance of each stage. Yeah. Yeah, good question. Yeah, go ahead.
Repolarization or
Yeah, so we get just initial repolarization in C, so just like a little bit of it. And then we get into plateau, and then it goes the rest of repolarization with E. Yeah, good question. Yeah. Okay, other questions? Yeah, go ahead. C, you said? E. I just said calcium closes, so the calcium channels close. Yeah, thank you. Other questions? Okay, so just for a sec with the people around you, talk about why that might be happening, right? Why do we have this slow building of tension in the heart? So this is like looking at the tension, the mechanical tension in the heart. It's building during that plateau phase, right? Just check in with each other on this for just a sec. Why do you think that might be the case?
Oh, yes.
This is right, it's just there.
It's like the initial polarization and then the plateau phase.
Yeah, sure.
When we were talking about this, is kind of off different,
Okay.
or the previous slide,
Yeah.
but in cardiac.
Yeah.
How we have the L-type calcium channel.
Yeah.
The I just had, is that the cardiac equivalent of the skeletal muscle by hydro?
In a way, but not completely. It is because it's the one on the membrane that's interacted with by the action potential, but it's not the same because it's not attached to the ryanodine receptors like the dihydropyridine receptors are. And the dihydropyridine receptors don't let calcium in. They just change from the outside.
Yeah.
They just change the ryanodine receptor so it can be let in from the inside. Yeah, that's good.
It's not attached.
It's not attached to that, yeah.
He said something here, the amount of calcium relies on the heart rate.
Mm-hmm. We'll kind of get in more contractility. We'll talk about that a little bit right now, but we'll talk about a lot more in the cardiac section. The next slide kind of gets into this a little bit. Yeah, yeah. Yeah.
So in B, the sodium influxes, is
OB,
there,
yeah.
like, is it sodium, potassium, ATPase pumps just working to build a gradient the entire time?
Yeah, always
Okay,
for
so-
sure. Yeah.
So the sodium is gonna get pumped in, but those pumps are gonna then be pumping sodium out.
Helps us recover from that, helps us recover from an action potential does, but this is so much faster than the pumps.
Okay.
That it just depolarizes quickly, but then, yeah, during, like from C then to E, the sodium potassium pumps are working to kind of help us
So you're
recover.
establishing the gradient.
Yeah,
Okay.
exactly.
That
Yep.
makes sense.
Mhm.
Yeah.
Yeah, good question. Yeah. Okay, let's come back together here. Okay. So any thoughts on that? Any thoughts on what's going on? Why is tension building there? Anybody? What'd you guys come up with? Yeah, go ahead. Uh-huh. Yeah, so we're giving it a chance to fill up a bit for sure. And this is the length of this is interesting. I mean, it's only 300 milliseconds on this x-axis, right? So it's not a ton of time. But if you compare that to a skeletal muscle action potential or a neuron action potential, those are about 3.5 milliseconds, right? So this is incredibly longer than that, right? So there is a little bit of filling that happens here for sure, and that stretches out our heart a little bit, which does help us elastically produce force. When we stretch out our heart, it elastically bounces back. And it also helps with the length tension relationship. Like we were just talking about in skeletal muscle, we put those sarcomeres of the cardiac myocytes in a good position to produce force, okay? But that's not the part related to this graph. Did anybody else have another thought they were thinking about? Yeah, go ahead.
It's all of the shortening.
Okay, the actin and the myosin, absolutely. And the reason that it's building more is because calcium's coming in, right? Calcium influx leaves us depolarized, right? Which is what causes that plateau. So its only job isn't to change the membrane potential or to keep the membrane potential high. It's also to go in and bind to troponin, right? That's what it's doing, right? So as it's going in, it's binding to troponin, which each time that happens, we get another myosin head that can bind to actin, right? Which that's what's producing force for us, right? So kind of has this double action or this double function where calcium's going in, which keeps us depolarized in that plateau phase, which allows for some filling time and all the things we're talking about, some tension building, but calcium also goes in and binds to troponin, and then we get more actin myosin binding during that period, right? So that gives us more ability to produce force there too. Okay. All right, questions on that? Okay, just a couple other things about cardiac force production. And we just kind of talked about all of these, so that's good. So the stretch of our heart is going to be dependent on how much we fill it up, on diastolic filling, right? Filling at rest. And that's kind of what's really called the Frank-Starling mechanism or Frank-Starling relationship, is that as we fill up our heart, so this is diastolic interventricular pressure, right? That's like how much pressure we have of blood inside our ventricles. We can see for just a really small increase in the amount of blood we got in our heart, it leads to a huge increase in tension development, right? So this steep part of this graph early on is really helpful for us as we start increasing our filling of our heart, gives us a lot more force production. That has to do with two things, the things we already mentioned, the stretch of the heart, the fact that it's elastic, so we stretch it out and it bounces back. And then the second thing is that we're putting those cardiac myocytes in an optimal position to produce force based on that length-tension relationship, right? But then if we start stretching out our heart too much, we kind of start getting less bang for our buck there, right? Too much stretch of it doesn't really produce a lot of force because it's just kind of overstretched, right? And that tension starts going down for the amount of blood that we put in it. Okay, and that's kind of what's being explained there. Okay, sympathetic activation of our heart is via beta 2 adrenergic receptors. We'll talk more about those receptors later. But this is epinephrine and norepinephrine binding to these receptors, right? We can see that here. So epinephrine and norepinephrine binding to these receptors. You can see that's a G protein coupled mechanism, right? That's cyclic AMP, protein kinase A, those are the things we were talking about last time. The eventual, the end goal of that is to open up more calcium channels and to activate that circa pump that gets our calcium back into our sarcoplasmic reticulum, okay? So this would lead to increased force because of actin-myosin binding. And increased relaxation. The more we activate those circa pumps, the faster we pump calcium back into the sarcoplasmic reticulum, and then the faster our heart can relax so we can fill it up, right? One of the types of heart failure is diastolic heart failure, right? That's when our heart doesn't stretch back out and we can't get any blood in it, right? If you can't get blood in your heart, then you can't produce good cardiac output, right? So we can have, so the sympathetic activation makes us contract harder, but it also makes us relax faster too by activating all those calcium mechanisms in a more rapid way. We'll get into these mechanisms more when we get to the heart too. So don't get too hung up on these details for this test. Just know kind of the actions we talked about there. Okay. All right, just a couple things about smooth muscle before we finish out here. So one thing that's both true in smooth muscle and cardiac muscle is that we have kind of different ways that our nervous system interacts with our muscle cells. In skeletal muscle, it's always the end of a neuron going right to the muscle cell. In cardiac and smooth muscle, we have these neuroeffector junctions where we have lots of little kind of bumps in the neurons with a bunch of vesicles and mitochondria right there. And so we can release neurotransmitter along the whole line of the neuron, not just at the end, right? And so that's kind of a nice way when we have this big sheet of smooth muscle along a blood vessel or along our digestive tract, we can release neurotransmitter all along where that neuron interacts with it, okay? And yeah, so smooth muscle is going to be mostly the sympathetic nervous system that interacts with it, but we can have up to 20,000 of those little bumps, those varicosities in a single neuron. So lots and lots of opportunities to release neurotransmitter along the way. Yeah, cardiac muscle, we have both sympathetic and parasympathetic activation all throughout for the most part. We can, yeah, depending on the tissue we're talking about, this release of neurotransmitters can be excitatory or inhibitory, right? So we can cause relaxation of the smooth muscle or contraction depending on where we're talking about. Same thing with the cardiac muscle, depending on which neurotransmitter is released. Right? Okay, just a couple things about smooth muscle that makes it unique. We do have gap junctions, just like cardiac muscle, but not a lot of mitochondria. They're not big energy producers. Still got myosin and actin for sure. We have these structures called dense bodies, which are kind of these anchor points for the muscle that kind of is in these little filaments that wraps around the cell, and it kind of just pinches it down almost like a net, like around something. Right? It's just kind of like pinching the whole cell down. Some of these muscle cells are multi-unit, meaning that we have, that they're not connected to the other cells. These are like ones that we find in our eye, in our iris muscles. And then some are unitary, which are all interconnected, like these big sheets that line our blood vessels in our GI tract. We have kind of a couple different types, but the blood vessels, we have both of those types there. Okay, just real quick on this, and this was something you looked at for the quiz as well, so you've already seen it. But we don't have troponin in our smooth muscle cells, so it changes the mechanism a little bit of with how we activate things. So the action potential is going to directly lead to calcium influx, but instead of calcium going and binding to troponin, it binds to a different type of protein called calmodulin. And so we can see that here, that calmodulin protein, through a series of events, is going to activate this MLCK, which is myosin light chain kinase. Okay, so we can see that MLCK there. That myosin light chain kinase is what's going to cause the phosphorylation of the myosin head. Okay, so similar, we still need to phosphorylate the myosin head. We just have a different mechanism of doing it. Okay, that's going to lead to contraction and shortening. And then once we decrease that signal, then the calcium decreases. We decrease the activation of myosin light chain kinase and other mechanisms like nitric oxide and other things that are vasodilators, for example, will activate this other one, myosin light chain phosphatase, which pulls that phosphate off of the myosin and makes it relax. Okay, so just a slightly different mechanism, but still, gets the contraction happening, but no troponin, those are the main differences there. And let's just finish with this last slide, and we'll start next class with a little recap there. So the other thing that's interesting about smooth muscle contraction is most of the time we need it to just be contracted, right? It's not like we just relax it completely and then we contract it like we do some of our skeletal muscle. Even though skeletal muscle, we have tone in it as well. But tone, which is like constant contraction, is a more important concept in smooth muscle to maintain blood pressure in particular. And so we have this special protein called calponin, which is like another binding point of actin and myosin outside of the myosin head and get into actin. So when that myosin head attaches to actin, you can shorten it. But we also have this other binding point with calponin, which directly attaches those two. And that can just leave it kind of locked in place. And this is what's known as the latch bridge mechanism that kind of gives us this sustained contraction and tone that we get in smooth muscle. Okay. All right, guys, we made it. So just imagine another hour and a half of that. That's how it was the last 4 years, but I don't know. I'm tired. I'm not sure about you guys, but anyways, we're good. That's all we got, 10 o'clock. So have a great rest of your day. We'll see you guys tomorrow morning at 9:30. Yeah.